exam that we're talking about. Okay, so we'll talk about the ear. And this is one of the, hearing is one of the special senses that we talked about. One thing I didn't talk about last time too much, because we're just doing the eye, which is pretty complicated, is this whole idea of receptors. And you have to go back to your nervous system lectures. Now, WCC does nervous system in A&P1, so you have a good idea what receptors are. So receptors are what senses the stimuli. So for the eye, the receptor was a photoreceptor, a rod or a cone. For, say, taste. Taste is your taste buds, papilla, have taste buds within them that are receptors, but they're chemical receptors or chemoreceptors. So taste, what you taste, the chemicals in the food, and I don't mean like chemicals they use to make fake food, but the molecules, I should say, in the food stimulate the taste buds for taste, like sugar or salt. So that's chemoreceptor. Again, the vision is a photoreceptor. And smell, which is called olfaction, you learn the olfactory nerve, which is a cranial nerve. Olfaction is also a chemoreceptor. That's for chemicals in the air, molecules in the air that we cognate in our brain as smell. Now, hearing, the ear, is not just hearing, of course, it's hearing and the balance. What keeps you walking straight? What keeps you from falling over? And that's what the balance or equilibrium is. So this is mechanical receptors, mechanical or mechano receptor. And that's really for vibration, is the stimulus. Okay, and there's fluid in these receptor channels, not in the receptor, but in the channels in the ear that sense the vibration that comes in from sound, or the movement that comes in when you're on a elevator, or you're walking down the stairs, or you stop short in your car, or just standing still, right? We need to maintain that balance or equilibrium. And we really call that vestibular. That's the word I like, vestibular. So if you hear the word vestibular, that has to do with balance, equilibrium overall. And it's sensed in your ear by mechanoreceptors and corrected and cognated in your brain. Hearing, there's a word called cochlear. And you'll see a structure called the cochlea for hearing. So that's important. And the nerve, that's really important for us to cognate what we hear or how we balance, is the cranial nerve, the 8th cranial nerve 8. And that's a 2-branching nerve called the vestibulocochlear. Makes sense, right? All this should make sense. Nerve, and it's a cranial nerve, CN, and it emanates from the medulla oblongata of the brainstem. So it has a nucleus that correlates the sound, correlates the balance, and it sends a signal to our cerebral cortex. You remember the cerebral cortex is where we cognate things, where we know what we taste, what we see, what we smell, what we hear, and our balance. Are we balanced? You can get dizzy, right? Does anybody know, what's the term called when you feel lightheaded or dizzy? Anybody remember it begins with a V?
Is it vertigo?
Yeah, vertigo. So vertigo is a symptom or a sign that something could be wrong with your vestibular system in your ear, in your inner ear, actually. So vertigo is pathological, so it says something's wrong. There could be something wrong with the vestibulocochlear nerve. It could be something wrong with back in the brain, or it could be something wrong in the ear. But of course, you have to find that out because vertigo could be has many other causes, not just inner ear issues. So it's inner ear dysfunction or something's going wrong with those, which you're going to see all the little parts inside the ear. So these are big highlights here. Know the receptor, know the nerve. Receptor is a mechanoreceptor for vibration. The nerve that brings the information, which is special sensory nerve, right? Special sensory nerve. It's not motor. It's not going to send a signal back to move the ear or anything like that. But there are little muscles in the ear you're going to see. Then there's skeletal muscles that do move, but it's more reflexive, even though it is skeletal muscle, which is really interesting. Okay, so go through this PowerPoint and just answer all these questions. And on Blackboard, I put a list of everything, like a checklist for the exam, so you can cover all bases when you're preparing. Okay, so we'll talk about the bones are called ossicles in the middle ear. You'll see that. We'll see the outer ear. You'll see this is a really difficult area, the labyrinth that's in the inner ear. It's a very complicated area. And even now in science and medicine, we're still, having trouble treating problems in the cochlea, which we'll see for hearing. So deafness, of course, is a very scary thing. And sometimes it's nerve related. Sometimes it's conduction related. There's a lot of different things. And you'll see that when you do the lab. There's some pretty good animations in anatomy and physiology revealed. And we could trace the path of sound, like I've been saying, from the receptor to the brain, where we know what we hear or how we feel as far as dizziness or balance, vestibular. And this is vibration, so you're going to hear frequency. Like frequency is wavelengths. Like if it comes into your ear at this wavelength, rather slow, but it can come in at higher pitch, faster into your ear. Let me see if I can draw an ear, right? So there's different frequency. And then you have intensity. You might have heard of loudness or decibels, right? Decibels. There's a small b. Decibels is how loud something is. Like an airplane could be 120 decibels. And 0 decibels is quiet, right? Traffic could be like 60 decibels. Your lawnmower could be 80 decibels. So usually above 80 is damaging to the ear. So that's the intensity. The frequency is more the pitch, which you'll see. Intensity is more loudness. I like loudness. Everybody can understand that if their hearing is decent. Again, pathways, the structures, and that's really what we do in lab. And you don't get this in A&P lecture, so we do have to talk a little bit about the function, the physiology, but the structures is what I'm going to ask on the exams, on the practicals, if you will, if you want to call them that. You're going to hear a lot of new words like macula. You're going to hear vestibule, and then utricle and saccule, and we'll get to that. So those are the hard things. The terminology is a little new, and you'll never say these words again, like pretty much, unless you go into ENT, ear, nose, and throat, where you study the ear. So you'll learn them for the practical, and hopefully you'll remember that if you hear the word utricle or saccule, it has something to do with the ear, and it has something to do with vestibular function in the inner ear. And you see these semicircular ducts and canals, which also has to do with rotational balance. There's different ways. We move in different planes, right? Some days we're driving our car in an elevator, and some days we're on a ride at Six Flags or spinning around. So your brain has to write that based on what the ear tells it to do. So it's a special sense, sensory. It's not a somatic sense. Like I didn't mention a lot of that because again, you get that in A&P1. Always remember somatic senses are like touch and temperature that you feel, tickle, pressure, vibration with your fingertips. If you're, if you're hot, if you're cold, if you're tingling, if something's sharp, if something's dull, that fine touch is all somatic senses. You have visceral senses too. Like if you could feel overstretching in your stomach or your intestines, or ischemia in your heart, where your heart is not getting proper blood supply. They're all visceral senses, but these are special senses. And just to repeat them, We have the vision sense. We have, which is your eyes, which we studied last week with the photoreceptors, and cranial nerve 2, optic nerve. Then you have olfaction, which is an old sense. It's not even in the midbrain. This is up in the frontal lobe of the brain. Olfactory is the sense of smell. Remember, chemoreceptors, cranial nerve 1. And then you have gustation, which is taste, your taste buds, your gustatory senses. And there's different ones. You have the sweet, sour, bitter, umami, right? The savory. And there's multiple cranial nerves for this special sense. You have cranial nerve 7, facial nerve, cranial nerve 9, glossopharyngeal, and cranial nerve 10, vagus nerve, for special sensory for taste. Then you have the ear and vestibular, which is cochlear and vestibular. I'm sorry, they're both cochlear and vestibular. So that's your 5 special senses. So see now the parts, the easy stuff, especially the outer part, because you see auricle means ear, really, and it's that funnel shape. It's like a satellite dish to kind of funnel that sound into your external acoustic meatus or auditory meatus, either one. And that's the outside. Remember the internal acoustic meatus from skeletal labs? Well, that's what houses the vestibular cochlear nerve, which is 2 branches, right? Cranial nerve 8 has 2 branches, a vestibular branch for balance and a cochlear branch for hearing. So the external acoustic meatus is part of the external ear, or you can say outer. Sometimes that helps. Outer ear, two T's, right? So that extends inside. Now, the tympanic membrane is your eardrum, and they're glands, and these, you get wax in your ear. The wax is called cerumen. It's a lipid, and it's really there for protection. So the glands that produce it, of course, ceruminous glands, they're epithelial tissue that are glandular, that secrete this waxy lipid called cerumen, and it's there to protect. You have it, first of all, it protects from things getting in. It's a first line of defense, really, of things getting into your ear, like bacteria. It also has, some chemicals that are antibacterial. So, of course, it doesn't want to get too much water in. It doesn't want to get dust in. It doesn't want to get viral or bacterial agents, which are microorganisms. So it has a function, not just to get it cleaned out. Like they say, you shouldn't put anything in your ear except for your elbow. So that means you shouldn't put anything in there. But we use Q-tips all the time, right? Because it's delightful. We get to clean out our ears and actually hear a little better sometimes. Then you have the tympanic membrane, which is also known as the eardrum. And that pretty much separates the outer ear from the middle ear. So there's 3 parts. There's an outer ear, middle ear, and an inner ear. And it gets more complicated, of course, as you get to the inner ear. So this is kind of like a little funnel. It does look like a funnel. It's epithelial tissue. And it vibrates and sends a signal, a vibratory signal to the middle ear. Here's some pictures. This is a good one. So this is kind of what I'd show you, Tariq. Like I would, you get this picture. There'd be multiple things we could ask here. So let's just start with the outer ear. Sometimes they call this auricle, or they call this part, which is elastic connective tissue, we call that the pinna. But the whole ear is called the auricle, all these parts, all this connective tissue with epithelial tissue covering it. And here's the external acoustic or auditory meatus. Meatus is just a big canal, a big cave. And then you have your tympanic membrane, your eardrum, right here. And right behind that is this kind of strange area. I don't think I would ask you this, but the tympanic cavity, because you really don't know what that is. It's just the opening. Okay. And then that's all external ear or outer ear, all of this. And you can see this is part of the temporal bone right here. This is all petrous portion of the temporal bone. See that thick portion of the temporal bone that houses these they're very delicate structures, really. They're bones, but inside you have tubes. I just want to give you an overall view. We're going to go through all of these. But this is the cochlea right here, just to give you some general view. This is the inner ear. And this is the semicircular canals. Then you have what's called a vestibule in here. The middle ear has 3 bones and muscles that move these bones. And we'll go through those. But this bony labyrinth, this is pretty interesting because it's a bone structure on the outside, and it has like fluid underneath it. And there's kind of like a membranous tube that runs throughout this bony labyrinth. And it's kind of like a bicycle tire. I don't know if you guys have ever seen that, where your tire on the outside is that rubber, and the inside is a tube, an actual tube filled with air. So it's kind of like that, with fluid outside the tube and fluid inside the tube that transmits the vibration to the receptors, which are deep in the middle ear. It's very complicated. And then you can see your nerve, vestibular cochlear nerve, going through the internal acoustic meatus en route to your brain, to the temporal lobe of your brain. And it makes a couple of stops before there. Now, again, you have to kind of remember some of the things you learned in nervous system in AMP1, which was a pretty complicated part of this class, I have to say. I remember the vestibular cochlear nerve has 2 branches, the cochlear branch denoting sound, and the vestibular branch denoting your balance. Okay, so here's the vestibule. You see these are just bringing big parts to you. And you'll kind of memorize the 3 auditory ossicles. They're the tiniest bones in your body. And there's muscles attached to them, which are the tiniest muscles in your body, skeletal muscles. And they have joints between them. It's crazy. It's like a regular joint system. It's some really strange design. So the middle ear has the big things of the bones, the ossicles, the malleus, the incus, and the stapes. The malleus looks like a hammer beating from the tympanic membrane, the eardrum. The incus is kind of like an anvil, if you know what that is. I'll show you again. And then the last one that comes into contact with the vestibule is the stapes, which looks like a stirrup. And then the connection between the stapes and the inner ear is the oval window. That's hard to remember because it's a round window, which we really don't see till later. Then in the middle ear, you have this canal right here, a canal that's going to your pharynx, to your throat, really, your nasopharynx, right behind your nasal cavity. And that's called the auditory tube. You might have heard this being called the eustachian tube. It's the same thing, auditory tube. So we'll talk about that, why that's there, and why it equalizes pressure from the ear to the throat. So you don't want too much pressure in the middle ear because it could really cause a problem, destroy some of the structures, and affect hearing. So this semicircular canals, you saw that. So here, this is a very good picture to study, Tariq, when you're right before the practical, so you could see all the things we could ask about the ear. It's all right here, pretty much. On the outer stuff, now we have to get deep into the cochlea and deep into the semicircular canals and vestibule. Now that's really microscopic anatomy and physiology that I'm not sure if everybody knows what's there. But you should know this is the temporal bone. All this is temporal bone. Right, the mastoid process would be back here, but you have the temporal bone here, all on the side, because the ear is on the lateral cranium. So hopefully this all makes sense to you, based on what you already know. So it's kind of like a review until we get deep inside, of course. So this is pretty good. This is a pretty good PowerPoint. It tells you all the different parts, so it's good to study. Middle ear. Okay, it has the tympanic cavity, which is internal to the tympanic membrane. So there's big bony walls in here. Remember, temporal bone. And it's actually the petrous portion. And don't forget, there's 2 ears. I don't want to not belabor the obvious, but of course, this is happening on both sides, completely symmetrical. But the strange thing is, you probably know, you could have issues with hearing on one side and not the other, which happens a lot. You could have one ear causing problems in your balance. So they work completely independent, of course. And your right ear could be worse than your left ear or vice versa. Okay? Okay, so there's 2 openings. The oval window is where the stapes, the last ossicle, is coming in contact with the inner ear. So you saw that before, and we'll go back. Round window is actually further down the labyrinth, and we'll see that later on. So know that the oval window is what the stapes comes into contact with right now. The round window, really not sure exactly why that was put there, but it's probably for some rebound of vibration, kind of like a trampoline. It has that kind of stretch effect, even though it's not that elastic, but it has like a stretch effect, kind of rebounds the vibration to further bring those vibratory frequencies to the receptors. Remember, the receptors are mechanical receptors, really pretty intense stuff in the ear. Here's the auditory tube, also known as the eustachian tube, spelled different ways in different places. And you might have heard of this. It's between the middle ear, that's what you need to know, and the nasopharynx, which is your throat. So this you might have felt like if you go in a plane and you're raising up, you're increasing altitude, the pressure changes. If you ever went diving, deep sea diving, 100-meter diving, as you go down, this pressure has to equalize between your middle ear and the atmosphere. And that, remember, when you go into a room or like one of those revolving doors where there's like a vacuum pressure, your ears kind of get really clogged for a second, and then they pop. Well, the auditory or eustachian tube is what equalizes that pressure, causes that pop. They tell you to chew, and because you chew because you're using your upper throat to kind of manipulate that tube into a good position where you can get that air. It's really air and pressure. Air has pressure, which can be painful, actually, especially if you're deep sea diving, if you've ever done that. So, yawning is one of the reasons we yawn is to equalize that pressure between the middle ear and the atmosphere, the tympanic membrane, really. So in kids, like I don't know if they do this so much anymore, but the auditory tube should be kind of like vertical, right? Like this, coming downward. So the pressures can, go back and forth due to gravity. But sometimes, like in children, they're too horizontal. They're too much like this. So if you get like a throat infection, like a strep throat, you're more likely to get a middle ear infection. That's why kids used to get a lot. I don't know if they still do. I still hear about it. They get middle ear infections because of the angle, and sometimes they have to actually take the tube. I would take this tube and install new tubes. So they get the tubes redone. That's what they call it. Have the tubes done as a kid. Sometimes even babies get this done just to prevent middle ear infections, which are extremely painful. It can cause hearing loss, ultimately. So it's the eustachian tube that they were working on. Here's the auditory ossicles. Now we're still in the middle ear. All right, remember, middle, external or outer, and inner. So middle is where the bones are. And there's the malleus, is the first one coming from the outside. So that's in contact with the eardrum, tympanic membrane. And they call it malleus because it looks like a mallet or a hammer. Tiny, tiny bone. Again, if you actually did a dissection, I don't think we could hardly find it. We'd have to use a dissecting microscope. The incus is the one in the middle. So the malleus will move based on what the tympanic membrane is doing. And then that'll come in contact with the incus. Now, this looks like an anvil. I don't know if you know what an anvil looks like. They used to forge metal on these things. They'd put like a sword on top, and then they heat it and forge it and pound it to looks like a pretty sword. Unless you watched Roadrunner or Looney Tunes, and remember Wile E. Coyote used to get the crap beat out of him. And then the Roadrunner would throw a big anvil that weighed about 100 pounds on his head from a cliff. So that's my remembering an anvil. So an anvil is just an old-time structure. So just say incus. It's a lot easier that way, right? And then stapes, it really looks like a stirrup of a saddle. And I don't know who designs this when you think about how this is designed for your DNA and carved out genetically to carve these bones using osteoclasts to actually make these little bones. It's crazy. And here's what you should know about the stapes. The stapes is the most internal bone of the middle ear, and it's in contact with the oval window. So now all this vibration that came from the outside went into the auricle, went through the external acoustic meatus to the tympanic membrane, into the tympanic cavity. Now you're in the middle ear, to the malleus, to the incus, to the stapes, to the oval window. So that's the transduction of this vibratory sound, which comes in frequency, measured in hertz. And that has wavelength, right? There's wavelength. So the shorter the wavelength, the higher the frequency. This is the wavelength between that frequency. Okay, and the higher the frequency, the higher the pitch, which you're going to see. The lower the frequency, the lower the pitch, right? Oh, auditory ossicles continued. Here we go. They're amplifying sound. Makes sense, right? It's like a built-in speaker in your ear. Kind of like when they try to reproduce this when they use things like hearing aids. So again, vibration, right? Coming from this frequency of vibratory sounds. Mechanical sound. It's going to a mechanical receptor, ultimately. We haven't gotten there yet. So the stapes in and out of the oval window with that foot particle. And it initiates pressure waves. Now we're going to the inner ears soon. And the inner ears, the labyrinth, cochlea, semicircular canals, and vestibule that are all fluid filled. Let me just tell you what the fluid is. The outer fluid underneath the bony labyrinth is called perilymph. I don't know why they call it lymph. It's got a lot of potassium in it though. And then there's endolymph. That's inner, in the tube. The interesting thing here, it's very high potassium, which is a positive ion. Now you got to go back to depolarization. Now this is nervous system. Now this is all under nervous system. Like when you go to your anatomy and physiology revealed, you have to use the nervous system module just to get into these two senses that you studied, the ear and the eye. They're the only two special senses that we're doing here in lab. So really get into this. So potassium is high outside the cell. This is completely different than your normal neuron cells and your other cells, muscle cells. So potassium is big. I think I need you to remember that. So there's two small muscles that restrict the ossicle movement during loud sound so it doesn't break anything, right? When you have loudness with high decibels, it just rocks your ear world. And you don't want to break those structures or destroy the inner ear because then you're going to have deafness. So remember I said, I think like an airplane is like 120 decibels. You can't listen to that every day. And if you work at an airport, you better be wearing protection, ear protection. Okay, so here's the two little muscles. Let's remember these. Tensor tympani and the stapes, stapedius. So the tensor tympani is attaching the malleus, and stapedius attaches the stapes. So stapes is closer to the inner ear, and the malleus is closer to the outer ear. By the way, a little trivia here. Stapedius muscle is the smallest skeletal muscle in your body. And between these bones, the malleus, incus, and stapes, it's actual synovial joints. Did you hear that? Synovial joints, like we have in our shoulders, our knees, our hips, our ankles, and elbows, fingers. So you can get arthritis in those joints in your ear, which obviously is going to give you a problem with conducting that vibration and affect your hearing. Arthritic ear. So as you get older, probably why you're most likely going to have some hearing loss if you really measured it. What'd you say? Huh? Okay. So here's a nice close-up. How tiny those muscles are, right? These are tiny muscles. Tensor tympani is a little bit longer, and they cut it right there. So this is a nice close-up of the eardrum, of the tympanic membrane. Remember, this is all outer ear. You have the big pinna right here, and the sound comes in, the vibration comes in, right? Boom, 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 bounces into the tympanic membrane, and then the malleus, which is like a hammer, takes over that conduction, and then the incus and the stapes, synovial joints. And you have these muscles that protect from over movement of these bones. Like this, look how tiny this muscle is. Look how tiny that is, man. That's the smallest skeletal muscle in your body. And then you have the tensor tympani, which is a little bit bigger and longer, probably does more work. Okay, and so now we're basically in the middle ear. Right here, here's the oval window and the foot process of the stapes coming in contact. And you can see back in the background, blurred out is the semicircular canals, the vestibule, and you can't see the cochlea. And the round window is actually a protrusion right here that really is part of the inner ear, because the inner ear is connected to that, those tubes, those labyrinths, which you'll see. Again, as we get deeper into the ear, the histology gets smaller and smaller. This is not so bad. We can see this kind of clearly, but it gets a little crazy. Okay, so now let's get into it. Now we're getting inside the inner ear, okay, beyond the oval window. And again, within the petrous portion of the temporal bone. It's really housed very safely in there for protection. Now the bony labyrinth, like if you pulled out that structure, the outer part is all bony, like a shell. It looks like a snail, right? With the whole thing is covered in bone. And then inside the bone, you have membranes. So it's maze-like, it's circular. It's very hard to understand the histology because it curls up. So the way the book gives it is not that easy to follow. So let's go. I'll tell you what exactly you need to know so you don't get too confused. So the fluid is called perilymph. So that's outside the tube. It's like between the bone and the tube, perilymph, which is really important. It could act as protection. It provides the potassium, again, which you'll see, to the endolymph. And then, so the outer is bony, the inner is membranous. Now, this is the tube, like I said, the bicycle tube within the tire. You know, how could I show you this? Like say this is the tire, the rubber part of your bicycle tire, right? And then within that, let me change the color, that'll help. Within that is an inner tube. So the outer tube is bony, the inner tube is membranous, like tissue, epithelial type tissue. So the perilymph will be between the bone and the tube in here. The endolymph will be in the tube, membranous tube. So membranes, right? Very delicate tissue here, by the way. So the endolymph, which is really what's going to come in contact ultimately with the receptor, is rich in potassium, which is a positive ion. You have to understand depolarization, right? Polarization is when you have a lot of positive on one side, a lot of negative charges on the other. So that's polarized, positive on one side, negative on the other. So if you allow the positive to come in to make this more positive, that's depolarization. And then you get action potential, and then we can get a signal to the brain. So you need depolarization to have an action potential to actually know what we're hearing or how we feel as far as vestibular. You need action potential. If you don't get that action potential, you're going to be deaf. Very important, very important. And you know most of the structures so far. So now we're still in the inner ear, and here this is the superstars of the inner ear. The cochlea is about hearing, and that's the snail-looking thing. Then you have these big semicircular canals. You have like one that comes up for the vertical movement. Then you have one that kind of comes in the middle, and then you have one that's horizontal. So they kind of have all those different moves: vertical movement, horizontal movement, and oblique type of movement. It's all about rotation. It's all about rotation. The vestibule is more about static, and that's like going up and down in an elevator or going back and forth in your car. So the vestibule is more about that. And when the vestibule contains those two parts called the utricle and the saccule, right? One's going to be for vertical, the other one's going to be for that horizontal movement. to keep you in check, to keep you balanced so you don't get so much vertigo that you can't stand up. So it's really important to know where you are in space. So here we are, a little bit deeper in here now. And I think these are really complicated, but again, I'm probably not going to put this on, like these pictures, because I'm getting confused when you look at it. But you have to know the terms. You have to know the general picture, right? So this is all in the inner ear, all of this. And this is the semicircular canals. And they're covered by bone. Everything's covered by bone, right? Here's your vestibule, which contains the saccule and utricle. So you should know those two. And they're in the vestibule. And they're for static equilibrium, whereas the semicircular canals are more for rotational, dynamic equilibrium. And this will be, again, repeated a lot when you do your labs and go through everything. And here's the cochlea. That kind of reminds me of a snail, all of this here. And this is a continuation. It has perilymph, it has endolymph, and there's what's called a cochlear duct. You should know what the cochlear duct is, right? I don't know if we see a good picture of it here. Cochlear duct, here it is, membranous labyrinth. So that's in the cochlea, the membranous labyrinth is basically the cochlear duct. And now we're getting to the receptors. We still haven't even gotten to the receptors yet. Like here's the cochlear branch of the cranial nerve 8. This is nerve, all right? And later on, you'll see the vestibular branch. So it might be hidden back here somewhere. So you have two branches of that nerve, and ultimately they come together when they go into the internal acoustic meatus to the brain. I'm just going to say brain now because it's a lot of things. Now, again, up here, I'm not going to go into this picture too much because, again, this is fluid filled. You understand what endolymph and perilymph is, basically. But down in here, the hearing is much more complicated. So deep within this cochlear, within the membranous tube, or labyrinth, you should say, is something called a spiral organ. This is key. I used to call it, as an old guy, I used to call it the organ of Corti. And this is key. This is where your receptors, now we're almost there. We're almost there, hang in there. I know there's a lot more left. But this is where the receptors are for hearing. And these are some really cool setup of receptors and how it relates that action potential into the temporal lobe of the cerebrum. So all about this fluid, you see the spiral organ coming in contact with the endolymph. Now the endolymph is transmitting vibration sounds that started on the outside, and it's conducted all the way deep into the endolymph. And ganglion is just a group of cell bodies that are outside the CNS, if you remember from AMP1 nervous system. So ultimately that's going to get all that information to the cochlear nerve, or the cochlear branch of the vestibular cochlear nerve. Okay. Otitis media, let's learn this. Otitis medius. See, this is middle ear that has bones, and usually otoliths are called bones. So otitis media, itis is always an infection. This is basically a middle ear infection, a fancy word for middle ear infection, otitis media. And this is what I told you before, where the young children, even babies and infants, have eustachian or auditory tubes that are too horizontal. So if you have something like strep, right, strep infection, which is a bacteria due to respiratory infection, which happens often, it could spread through the pharynx, the nasopharynx, through the auditory tube to the middle ear, which gets a lot of fluid inflammation because that's the reaction, the immune reaction to the streptococcus. And that could be painful. As appropriate as it is to try to destroy the bacteria, it just builds up and causes temporary hearing loss and hopefully not permanent hearing loss. So painful, right? This is, you don't know what's going on. There's fever, of course. Now, if you have strep, it's a fever, but it could be a viral infection too. Now, a strep infection would cause a high fever. A viral infection is a low-grade fever. So you have to decide, and then you do the histology, or you do the, you take a look at the microscope and you see what type of microorganism is causing this infection in the middle ear. And it came from the throat in this case. Here's a word for us, myringotomy. Myringotomy is that surgical procedure where they take the old tubes out and put in plastic new ones. I'm exaggerating, it could be, it's a very sensitive plastic, or I don't know the tissue they actually use or the materials they use, but they actually install new tubes that are more vertical, like the young kid has a horizontal, like I showed you. They take those out and install ones that are more vertical, where the fluid can drain back and be reabsorbed and rid and taken out of the body. Otoscope, that's the thing the doc puts in your ear to see. It's got a light source, and they could look in, they could see if there's inflammation, they could see redness, they could see pus. So the otoscope is the actual scope. I think I mentioned the ophthalmoscope last time with the eye and how you could see all the retina. So it's kind of similar to that. Sometimes it's just a matter of changing the head on the little tube with the batteries in it. Okay, so now again, we're getting deeper. Now I don't know how deep we're going to go here, but there's some key words I need you to know. Of course, cochlea. When you hear cochlea, you think inner ear, you think hearing. Bingo. That's the takeaway here. It looks like a snail, so visually, Tariq, you could see what that looks like. And then you have this modiolus. I probably won't ask you about that because it's just an axis within the organ. Cochlear duct is kind of important because now we're getting into the receptors. So the cochlear duct is just another name for that membranous labyrinth, the inner tube, if you will, that contains endolymph. And the top of it, I'm going to see pictures of this. Now you got to pay attention here. This gets a little crazy. The roof of the cochlear duct has what's called a vestibular membrane, and the bottom is basilar membrane. Then you have these chambers, the scalar vestibuli, scalar tympanate, and the helicotrema. I just like saying that word. Probably never ask you that on a test, but you see it in the lab. So these are chambers and channels within the cochlea. But really, I need to get to these membranes. I need to get to the receptors, get this vibratory conduction to these receptors. So here we go. Now the spiral organ, which I also call organ of Corti, this you have to know. So if I ask you where are the receptors of hearing, you have to say in the cochlea and part of the spiral organ. And that's the sensory structure. This is the nerve tissue. This is tiny, microscopic. It's a miracle we even found out about this. So here's the deal. These epithelium, some of it's thick, some of it's specialized, contain hair cells. Remember hair cells, used to call them the cilia? So the top of these receptor cells have cilia, hair cells. That's the receptor. Crazy, right? Right? You have inner hair cells and outer hair cells. And this is important too. On top of the hair cells, you have these long stereocilia. Now we're getting into microvilli, right? Microvilli. So stereocilia and one kinocilium. Kind of looks like this. You have your cell, then you have the cilia, stereocilia, and then you have the kinocilia on top. So the hair cells are this, with the nucleus. It's epithelial tissue, really. Stereocilia, like microvilli on top. And then you have the kinocilia here. So it's going to come in contact with a membrane that's going to have nerve tissue attached to it to send the signal from the hair cell, which is the receptor for the vibration, to this membrane, to the vestibular cochlear nerve, in this case, cochlear branch. And the membrane is called the tectoral membrane. Sounds right. It's sensing something. It's tecto means touch. So the tectoral membrane is really what these receptors are coming in contact to send the signal. And the spiral ganglion is the cell bodies of the cochlear branch of the vestibular cochlear nerve. And don't worry about modiolus. That's too complicated. So this is really intense how this happens. And we really don't have an answer for it. Treating deafness, We use hearing aids, but there's something called a cochlear implant. And I don't know if you've heard of that, but that's like putting in an artificial cochlea with all these sensitive nerve tissue. So now again, this is the computer, age, and we can definitely reproduce a lot of this, but it's a pretty unsuccessful surgery. Very few people have success with it, but it does work and is very hopeful to cure deafness, conductive deafness, or neurosensory deafness. It's more neural deafness can be helped with this. So again, there's a lot of crazy stuff. So here's a picture of all this. Let's get the real big parts here. You have the vestibular membrane and the basilar membrane, the scala vestibulari, the cochlear duct. Now that's basically just a fluid here. You can see that. It's not showing you the spiral organ in this picture. So this one I don't like too much. I mean, as far as naming these things, I don't like this one too much for you. This is a close-up. Now this I do like. Now this is the close-up. And here you can see the main organ where the receptors are, the spiral organ. And it has the tectoral membrane. This is key. And the basilar membrane on the bottom. Scala vestibularis on top, tympani on the bottom. Here's the spiral ganglion leading to the nerve, the cranial nerve 8. Very difficult here. So this is deep in the cochlea. You have to know spiral organ, really important. And the tectoral membrane, because that's where the information, the depolarization is going to happen. By the way, it's set up in here. This is kind of unclear too. Way back, I saw this. Where was this? We're talking about this depolarization. And there's a neurotransmitter released. Remember neurotransmitters? And neurotransmitter implies that there's going to be a receptor in these tectoral membrane. And most of the time, the neurotransmitters, we believe it's glutamate. I'm not even sure. I don't know if they're sure. So the glutamate has to be released from the hair cells to get a depolarization, to get an action potential. And I don't even know if everybody's sure. The scientists are sure of that yet. So, but it's a good guess. Okay, where were we? And that was a real close-up. First of all, I want to make sure you guys are okay. Are you guys okay? Are you with me?
Yep.
This is not easy.
Good.
This is not easy.
So,
Yeah. So I'm trying to, Vince, I'm trying to make this where you could just know the big parts. So again, I give you a checklist which has everything you need to know, what you need to identify, the little facts you need to know. So that'll help you. So, and I looked at the labs, the animations. They're pretty good. They're pretty straightforward too. So by the time the week is up, you should have a good idea of what all this means, even though it's a very complicated thing. That not even the scientists know 100% what's going on. The year is very, very crazy, right? It's pretty intense. Any questions before I go into the receptors now? We're almost there. We're getting there. Everybody cool?
Yeah.
Yep.
Okay, let's move on. Yeah, just feel
Yeah.
free to stop. Again, I'm recording this, so it's helpful, when you go back and listen, if you get a little lost now, as we all do, So now we're deep in the organ of Corti, the spiral organ, where the magic happens. And the fluid's coming through the cochlear duct into this area. So now you can see the inner hair cells and the outer hair cells. And then you can see, look, the stereocilia, remember that on top? And then you have the kinocilium on the top of the stereocilia. It's hairs, it's hair cells, and that's how we hear. It's how those hair cells bend on the tectoral membrane that releases neurotransmitter and sends a signal to the spiral ganglion and off to cranial nerve 8, which is vestibulocochlear nerve. In this case, it's the cochlear branch, though. This is the cochlear branch. So I think by now you have an idea that cochlea means, or cochlear means hearing. So that's a big help, And here it all is. And here's the tectoral membrane right on top. It's basically a mix between epithelial and nerve tissue. And that's the receptors right there. So technically the kinocilium to the tectoral membrane is your receptor for vibration, mechanical receptors, because they're bending, they're moving, right? The hair cells are moving, the stereocilia are moving, the kinocilia are moving and bending. So I think that's the best way to understand it. Keep it simple, right? The basilar membrane's on the bottom. Always remember that. The tectoral membrane is on the top of the hair cells, specialized cells. How could you reproduce this in a laboratory? I don't know. So here it is. This kind of gives you a pathway, kind of a recipe of how this all works. So here it is. Sound waves vibrate from the outer ear to the tympanic membrane. Then the ossicles, you should memorize them. Malleus, incus, and stapes to the oval window, which is the beginning of the inner ear. And it's fluid now. So this is another thing, kind of like the eye, where whatever the stimulus is, in this case, it's vibration coming in the ear, and then it goes from air to fluid. So now the fluid is good. The pressure in the fluid, the vibration in the fluid will continue into the scala vestibuli, and then the membrane, causing endolymph to change its frequency. It depends on the pitch, right? It depends on the wavelength of that vibration, of that sound in our case. And ultimately into the cochlear duct, which is endolymph. It's part of the membranous labyrinth. Okay? So the basilar membrane's on the bottom, and that moves. And then the hair cells are distorted. That means they change shape. And then you have a neurotransmitter release, which probably is glutamate. Okay, so that's going to start something, the neurotransmitter. And I'll tell you what it's going to start. It's going to allow potassium to enter the hair cells. And that's depolarization. Remember, positive. And this is not like regular nerve tissue, though. And that causes depolarization. Now we can get a signal to the brain. Okay? And this round window thing is kind of confusing. I think just the pressure of the endolymph kind of pushes against like a trampoline and propels it towards the organ of Corti, or the spiral organ. I'm sorry, I keep saying Corti. It's the same thing. So this shows you the steps. I like this too. So this is kind of what you'll see in the animations, which is really helpful. You'll see the whole, which basically what we just did, the sound waves coming this way, and one is coming into the middle ear, and the three, and they're called ossicles, right? Here's showing the amplification, pitch, and frequency. And then here's the nerve, and deep into the spiral organ. Basically what we just did. So this is kind of cool, but I think I have to stay with you, so make sure you go through. I'll check, make sure everybody's seen the videos, the animations, because again, it's complicated. But again, I think the practical is going to be a lot simpler than you think. Like telling you, just as long as you know the main parts, you're able to tell me, answer some of these basic questions, and all the parts being able to label. I mean, it's just the ear. Again, it's not a huge part of the practical, but it's part of it. So this is telling you again about ion channels. And again, I think you should know about potassium coming inside the cell, right? And that causes depolarization. And then they release a neurotransmitter. The cells release a neurotransmitter for action potential. And action potential means that that signal will get to the brain. So we know what we're hearing. That's really important. Now it's going to get even a little more complicated. This is not so much. This is just showing, this is actually pretty good. It shows you a close-up of the kinocilium and the stereocilia, really mostly stereocilia. And then they're kind of linked together. So they kind of move. The animation will show you them moving and kind of like wavy hairs. Can't see it here because this is a static picture. And this is the ion channels for potassium coming in. So it's really positive in the extracellular fluid. And then when the neurotransmitter binds, boom, depolarization. Then you get a nervous signal to the cochlear branch to the brain. So we know what we're hearing. So this is kind of cool. This stuff is now just kind of breaking down what actually you're going to hear. And sound perception. So you can't just pass that up. The perception is actually, okay, what did I just hear? How loud was that? Do I like the sound of that music? Is it Mozart? Is it Kanye West? Is it Machine Gun Kelly? Is it Jimi Hendrix? guitar? Is that a drum? Is that a flute with a higher pitch? Is that a high voice? Is that a low voice? Is it pleasing? Is it not pleasing? Does it cause a memory? Have I never heard that before? This is the perception of sound is part of life, right? And sometimes it could be a protective mechanism. If you hear a bang, shot, you have a reflex that'll turn your head towards that shot, right? And the pitch, you should know, depends on the frequency, how fast the waves are going, right? Hertz. So we could hear between a frequency of 20 to 20,000 hertz. Hertz is the frequency of how many waves over time. So, they say that dogs can hear at different frequencies, either higher frequencies or lower frequencies. So humans hear between 20 and 20,000 hertz. So you want to keep it pretty low, right, to save your ear. And there's different pitches, different keys. So again, you don't need to know this. This is too complicated, like about the stiff basilar membrane and all that. Just know there's high frequency and there's low frequency. And high frequency is high pitch, low frequency is low pitch. Okay, and that's what this is showing, the apex. I don't think you need to know. I'm not going to ask you this at all. Okay, loudness is important because they were talking about decibels. Oh, that's the way you write it, small d, big B. Okay, so threshold of hearing is 0, 0 decibels. It's nice. So you can't hear anything lower than 0, which doesn't make that much sense. So, but I would say the lower the decibel, around 80, above 80 is starting to get dangerous on a regular basis, like a lawnmower or a car horn. So they should be low to protect your ear. So larger movements, larger sound. And remember, we keep talking about the temporal lobe of the cortex. That's where we interpret it. Our cerebral cortex, cerebrum, temporal lobe of the brain now. Don't get that confused with temporal bone. Temporal lobe of the cerebrum, which is where we cognate sound. And there's a couple of stops in between that, which you might remember. And it's really exponential. Like if you hear something that's 40 decibels and it goes to 50, that increases 10 times more intensity. So an increase of 10 decibels is a lot. So be really careful with your earbuds when you're listening to music. You can get away with it for a while. It's pretty cool. But, ongoing decibel increase in your ears, it really can cause damage to all these really sensitive structures if you haven't seen this already. Crazy, right? And here's the nuclei in the brain. This is kind of important. Of course, cranial nerve 8, which is a peripheral nerve, right? Peripheral nerve. The cochlear nucleus is a group of cell bodies within the CNS. The ganglion was a group of cell bodies outside the CNS. And it's in the medulla oblongata, which is part of the brainstem. That's not where we think. The temporal cerebral cortex is where we cognate and think about what we hear. So remember the midbrain, right? This inferior colliculus of the corpora quadrigemina, that was where we reflexively respond to sound. Like the superior colliculus is for vision. The inferior colliculus is for sound. And then the midbrain to the superior olivary nucleus. Now you're not going to really have to know that, but I like this inferior colliculus thing because we had that in AP1. And then it goes to the thalamus, where all sensory information is processed, besides smell, to the medial geniculate nucleus of the thalamus. Now, that's part of the diencephalon. Now, with vision, it was the lateral geniculate nucleus. So this is kind of important because this is a reflex. Remember I said before, like if it's protective. So if you hear a sound to your right, like a snowplow just dropped its plow, I turn my head to the left really quick. But I don't have to think about doing that. It's reflexive. So this pathway between the medulla oblongata, the inferior colliculus of the midbrain, and the medial geniculate nucleus of the thalamus is going to send a reflexive signal back to your sternocleidomastoid muscle in your trapezius to turn your head towards that sound. It's a protective mechanism. That's what a reflex is. So I said reflex. This is the cortex. Cortex is the temporal lobe, of course. And you should know that. I think that's the main takeaway here. And here it is. Again, you have to kind of go back to your nervous system. This is an A&P2. We do special senses, like I said last time. So the cortex, the cerebral cortex, that outer 5 to 7 millimeters of gray matter is where we do our thinking and perceiving and initiating movement. So the part of the cerebral cortex that's involved in hearing and vestibular is the temporal lobe. Really important. Okay. And this shows you the pathway. See, I love this stuff. Again, you might say, oh my God, back to the brain. Here we go. But this is kind of simplified. And let's, you know, you can read through this, but let's see what happens. Like, and we didn't even talk about vestibular yet. So the cochlear part, the hearing, it's all sensory. So it's all afferent coming into the CNS. You know, it goes through this actual synapse here in the midbrain. And then up, I'm sorry, into medulla oblongata to the midbrain. And also the cerebellum is involved in this too, because you know, as far as movement and balance, which you'll see later. And then it ascends, and this is the thalamus, the medial geniculate nucleus. Down here is the superior colliculus down here. And then it goes to the cortex of the temporal lobe where we cognate it. Great. I have no idea how this works as far as in here, where this comes from, how we feel and see. Just with this gray matter that's 5 to 7 millimeters of cortex, all of this, but this happens to be the temporal lobe. And it's going to be the same basic area, except different pathways for vestibular. Okay, so you can have deafness, you can have conductive deafness, which is more like a block of the vibration. Like you can't get that vibration to the spiral organ for blockages. And I mean, you could have temporary conductive deafness with too much wax or fluid in your ear or the infection and all that, virus, bacteria. So it's interference of the wave of vibration. Sensorineural is a problem with the nerve. And you should know this, the two difference. This is more a mechanical problem, where this is more neurological problem. Sensorineural, because it has to do with the nerve. It could have something to do with the receptors too, or the potassium, or the neurotransmitter, or the tectoral membrane, or the kinocilium. All that's involved in conduction of these nerves and depolarization to action potential. Okay, so let's take a break. I think this is a lot, and equilibrium is not, it's pretty intense too. So what I think you guys should do now is actually go to the lab files. Let's go over to, I don't know if you could see what I do. Stay with me for a minute, guys, so you could tell me if you could see this stuff. Now this is, let me share my screen here, because this is really important. I don't know if I could share the screen where you could see these animations together, or you need to go off on your own and do them. Either way, you got to do them. So everybody see this? Just let me know if you're awake, please. I know it's a tough day.
Yeah, we can see it.
So what you're going to do, like, I'm going to go back to equilibrium in a bit. I'll tell you what time. You have the ear, and because these are only, I think the biggest animation is like 4 minutes. So we did the PowerPoint so far for hearing, but I think you guys should go here to these first before we move on. So, oh, well, I have to log in. Why do I have to log in? Probably because I have one open. Anyway, that should take you right to the video. So could you try that for me right now? And you have to do 4 things.
Yeah, it worked for me.
Are you able to access that animation? Or this animation, I believe that one.
The hearing one, yeah.
Cool. And let me see another one here. I don't know why it's asking me to log in. Probably because I'm logged out, because McGraw-Hill like times you out very quick. Everybody notice that? It times out really quick. So you might have to go back and close stuff and come back in. So you're going to do this hearing. You're going to do hearing loss, 3D. You're going to look at the ear dissection. That one you don't have to spend too much time with now. And then the last one is here. And then we'll do the practice models later. But for now, take, what time do we finish? At 10:50? So we'll come back at 9:45. And we'll do equilibrium. So go right now, go to those 2 videos and just go through them. I'll test to see, I'll track the statistics and see that you're doing it. Or I mean, if you don't want to do it, take a break and do it later. That's fine. I mean, I don't know what to tell you about, when we're not in the lab. So go in and take a look at that, and we'll come back at 9:45. Either way, you need a break because I've been yapping for an hour and 15 minutes. So sorry, Todd. So we'll come back and I'll talk to you about equilibrium, and then you can go into that. I really like the animation, so I wish I could watch them with you again, but I got to figure that out how to do it. But do it on your own. And if you don't want to do it now, you can do it later. I'd rather you do it now. So, because you've taken this time every Monday morning at 8 o'clock. So we might as well do it. Then you might have questions for me. Probably not, because they're really good. So come back at 9:45. That should be plenty of time. And I'll do my best to get you out of here with the equilibrium stuff. So I'll see you guys at 9:45. Enjoy the videos. And no Looney Tunes. Don't watch the Roadrunner or Tom and Jerry. But if you want to, I'm going to watch it. So I'll talk to you about that later.